This is just a reiteration of the presentations for today. I'm going to start with talking about essentially where arsenic comes from and what are some of the basic issues that we have to deal with regard to this risk assessment. Arsenic, unlike most of the things that we have to deal with, is a natural substance. This is not a substance that any of you manufacture. This is not something that you go and add to your foods or water or anything. This is a natural substance that, for the most part, is in our environment because of natural occurrences. Yes, there's trivial amounts that comes from pharmaceuticals, trivial amount from various pesticides, and even chemical warfare agents. Arsenic has long been used for chemical warfare beginning in World War I. But nevertheless, most of it, it was placed here naturally. It's part of the Earth's crust. As you can see here, if you look at the entire Earth, it's present in about 2 to 5 ppms. In the U.S., it ranges from less than 1 to about 100 parts per million. And the average is about 7.2. The only way you can basically avoid exposure to arsenic is that you have to stop eating, stop drinking, and stop breathing. Then you won't have any arsenic, because the arsenic is in the food, it's in the water, and it's in the air. And the question really comes down to, okay, if it's there all the time, what's a safe level? Because we know that it produces certain toxicities. Now, most notorious of this, of course, has been cancer. And it's been known for over a century that it's associated with skin cancer and also the benign skin changes that are well known for arsenic, and were actually first discovered from the use of various solutions that were developed and used as pharmaceutical agents back in the 1800s. So this is not a new phenomenon that we're dealing with. The skin cancers is one, but with the data that came out of Taiwan in the late 70s, early 80s, it became apparent that it also is associated with bladder cancer, also more recently with lung cancer, and there's some indication that it may be related to certain other types of cancer as well, such as kidney and liver. It also has become apparent that in addition to the benign skin changes that occur with arsenic, that there's some effects on other non-cancer toxicities as well, including ischemic heart disease, which has been given a very high priority by the National Academy, which Jim is going to talk about shortly, diabetes mellitus, and a whole bunch of other potential toxicities. So there's a great deal of concern about this, and the bottom line is we're all exposed to arsenic. We can't avoid arsenic, but what's a reasonable and safe level to have in our drinking water and in our food, as well as in the soil? So obviously this group is most interested in food. Now, in many parts of the world, drinking water is the major issue with regard to arsenic exposure, such as certain parts of Asia and certain parts of Latin America. In the U.S., there are parts of this country that are actually, we're in Bermuda, so parts of the U.S. that the arsenic levels actually do get above 100 parts per billion. But for the most part, the U.S. is considered a low arsenic level country, as is most of Europe, as is Japan, and many other countries. So a concern has really surfaced that maybe it's not from the water that we have to be worried about, but food exposure. And this is just a listing of some of the places that we have measured arsenic. And you'll notice that I've listed here both total arsenic, but also inorganic arsenic. And this is a very important distinction. Total arsenic includes all forms of arsenic, many of which are essentially inert, including a substance called arsenobutane or butane, or however you pronounce it. A number of other substances which I'll come to in a minute. The key that, as far as toxicity goes, though, is the exposure to inorganic arsenic, which is mainly arsenate or arsenite. And as you can see, for some things, most of the arsenic is actually inorganic. So juices, for example, have very low levels. This is an average, and this is from a report in the Schuf et al. Publication from about 1999, and hopefully Dr. Fitzpatrick will talk about more details of this in her presentation if we can connect with her. But you can see that most of the arsenic in juices is actually inorganic arsenic, so they're quite comparable. In contrast, rice, most of the arsenic that's present is not inorganic arsenic, so we have to pay real attention to what kind that is. Others is even greater disparity. So for example, seafood has enormous amounts of arsenic. If you want to get arsenic, go to the restaurant here and have some seafood, any seafood, whether it's fish, clams, crabs, whatever you want, it's going to have a lot of arsenic. But almost all of it is not inorganic arsenic. It's a basically an inert type of arsenic that we don't have to worry about toxicity. And again, the same thing just for comparison is beef, chicken, tomatoes, and on. Most of the arsenic in these foods is actually not the arsenic that we're concerned about. But nevertheless, the overall amount of inorganic arsenic that we're exposed to can become rather substantial depending on your overall diet and where in the world you're living. So I was mentioning that arsenic comes in many forms. The form that we're most interested in with regard to toxicity is inorganic arsenic, either arsenate or arsenite. And almost all the arsenate that you're exposed to is converted in the GI tract or in your bloodstream to arsenite. So you can interconvert them pretty closely, particularly in various studies. And most often these are not distinguished. They're just inorganic arsenic. But in addition, we do have all these organic forms, such as methylated arsenic, and there can be mono and di. In rodents, trimethyl arsenic is very prevalent. In humans, this is almost non-existent except for exposures that are essentially lethal, so we don't worry about the tri so much. Arsenosugars are a major component of a number of foods, particularly foods like rice. Arsenobetaine is a very common constituent of arsenic in seafood. And then we also have arsenolipids, arsenylcholine, and a number of others that are being discovered as the chemists are becoming better and better at identifying this. Now, one of the problems that we have to deal with in arsenic toxicity and risk assessment is assessing how much arsenic we're exposed to and how much of that is inorganic arsenic. And the analytical methods are not great. They're good, but there's a lot of confounding factors that have to be paid attention to. And so when you're analyzing the data, you have to ask what methods are they using for the analysis. This is particularly true in distinguishing the various organic forms, but even the inorganic forms can pose difficulties as well. Now, just to show you how complicated this can get, here is the metabolism of inorganic arsenic. And essentially it goes through a sequence of reductions from the +5 to the +3 oxidative state, then gets oxidatively methylated, and then gets reduced, methylated, reduced, methylated, reduced. Okay, essentially these we don't have to worry about in humans, and exposure to all of these occurs in the environment. The difficulty happens is that our body's response to different forms of arsenic is quite variable depending on whether it's exposed exogenously or we generate it endogenously. So for example, if you consume arsenate, you'll generate all of these 6 components in humans. If you consume MMA5, most of that will remain as MMA5. Some of it will be converted to DMA5, very little of it. The DMA5 turns out in humans, very little of it's converted to anything. It comes out as DMA5. Now this is of importance because one of the issues that we have with arsenic is exposure assessments. For all the epidemiology studies is how are you going to assess the overall exposure to arsenic. And some individuals are using DMA as a surrogate marker in the urine for arsenic, since that's the end stage metabolite for inorganic arsenic. The problem comes up The problem comes, and this is a slide from Dave Thomas at EPA, is that DMA5, which is right here, is actually the convergence point of metabolism from all these other forms of arsenic. So if you start with arsenyl sugars, arsenolipids, arsenobetane, arsenylcholine, or inorganic arsenic, you're going to end up with DMA5 as your urinary metabolite. And there's increasing evidence that in the U.S. population, that much of the DMA5 that we're measuring and using as a surrogate marker for inorganic arsenic actually does not come from inorganic arsenic. So it's complicating things a great deal, and this is something that's going to have to be looked at in greater detail. Now, just to back up a minute, the forms of arsenic, you'll notice, are either a +5 or a +3 oxidative state. The +5 oxidative state is essentially inert as far as biological activity. Except for the arsenate, the MMA5 and DMA5, for the most part, won't even cross cell membranes and does not react with proteins or anything else very much. In comparison, the trivalent, or +3 oxidative state, are very reactive compounds, and these interact with proteins and other macromolecules, and particularly sulfhydryl groups. Arsenic in the +3 oxidative state loves to react with sulfhydryl groups, whether glutathione or protein sulfhydryl groups. And this fundamentally is the way it interacts with cells. The one substance and one macromolecule that it does not interact with is DNA. That's been very conclusively shown actually by Steve Nesnow at the EPA laboratories that it does not interact with DNA, and chemically you can see why, because chemically there'd be no way for it to interact with DNA. So for cancer, which is my area of interest, there have fundamentally been suggested several possible modes of action. And I want to emphasize that the ultimate risk assessment for arsenic is going to have to come down to an understanding of mode of action. Because for us to extrapolate beyond the area where we actually have data at the high exposure levels, we're going to have to take into account the mode of action. Now, for cancer, clearly, if it's DNA reactive, like things like aromatic amines and nitrosamines, aflatoxin, these kinds of things, that's one mode of action. We know that that does not happen with arsenic. Any form of arsenic does not react with DNA. There's some evidence for indirect genotoxicity, which I'll come back to in a minute, including some evidence in human exposures, and that's been a source of some various articles over the last year that have raised certain amounts of anxiety amongst the regulatory agencies, including our own FDA. But also there's non-genotoxic mechanisms, which I'm going to focus on is cytotoxicity and regeneration versus direct mitogenicity. Now, as I pointed out, the only mode of action that can give you a non-threshold linear extrapolation for a cancer endpoint is DNA reactivity. And importantly, that is the mode of action that we can absolutely exclude as a mode of action for arsenic. So for cancer, really just based on basic principles of carcinogenesis, we can say that the carcinogenic activity of arsenic is not going to be linear, and it's going to involve a threshold. That's the source of the controversy. Okay, I mentioned that there might be some evidence for indirect genotoxicity, and this comes from assessments of so-called micronuclei in human populations, collecting urothelial cells in the urine and looking for micronuclei that are present. And what we were able to show that was in the mouse that these inclusions that occur, you can see them by electron microscopy here, accumulate in the mitochondria, and they've been mistaken as micronuclei in the cells. It turns out they're not micronuclei, and these then are cells, and you can see how degenerate they are from human urine. And again, you can see these exact same inclusions. These inclusions are what has been mistakenly called micronuclei in a number of epidemiology studies In fact, they're accumulations of inorganic arsenic bound to various macromolecules, probably protein and also some glycolipids and probably just complex sugars, polysaccharides. But the major point is that here's a picture of what's been claimed to be micronuclei from a group in India who we are now collaborating with. These are from patients that have been given enormous doses of arsenic trioxide in the treatment of promyelocytic leukemia. And you can see that by light microscopy, they're identical, and by electron microscopy, they're accumulation of these globules. This is essentially the same kind of inclusions that you see with lead in the kidney, and it's essentially a detoxifying sequestration of the compound in the cytoplasm that essentially is a way for the cell to deal with the toxicity of these agents. So what do we have with regard to mode of action and the evidence that it's actually cytotoxicity and not mitogenicity? Well, the initial findings with that came from research in our lab and that from Dr. Fukushima, who's in our audience here in Japan, that essentially dimethyl arsenic 5, let me go back here, is consumed, it's metabolized to the trivalent form, it causes urothelial cytotoxicity, there's regenerative proliferation, hyperplasia, and eventually tumors. And you'll notice that this slide is from the EPA, Vicky DeLarco provided it, and this is actually used as one of the teaching slides for mode of action for EPA for the pesticide branch in evaluating chemicals. And the nice part about this is the conclusion is this is a threshold mechanism. Now, that's for the organic arsenic. For inorganic arsenic, it turns out it's exactly the same thing. That you give the inorganic arsenic, and now it's metabolized not just to DMA3, but now we have to worry about inorganic arsenite, MMA3, and DMA3. It produces cytotoxicity, you get regenerative proliferation, and eventually you can get tumors. And there's a lot of evidence that this is how tumors are produced, not only in the animal models, but in humans as well, and I'll come back to that in a minute. Now, in vivo, in the animal models, it very clearly involves a threshold phenomenon. No matter what form of arsenic you administer, whether you administer it to rats or mice, whether you administer it in the diet or the drinking water, whether it's arsenite or arsenate, and no matter what your endpoint is for measuring this, whether histopathology or even genomics, the bottom line is the NOEL is 1 part per million. Across the board, multiple different studies, multiple different laboratories, it's always the NOEL is 1 ppm, sometimes a little higher than this, but that is essentially where we're at. In vitro, we can also assess this, and it turns out utilizing a variety of cell types, and these are the focus on epithelial cell types, and no matter what your endpoint is, the NOEL is somewhere around 0.1 to 0.2 micromolar concentration. So the real question then comes is, how much arsenic, inorganic arsenic, do you have to ingest to generate a tissue level that's at this level to produce a biological effect? Below that level, there's going to be no biological effect. Above that level, you're going to get a biological consequence depending on the cell type that it's interacting with. Well, it turns out to generate that kind of a tissue level is somewhere around 75 to 150 parts per billion of the drinking water, and presumably an equivalent amount in the food supply. Now, this is well above the 10 ppb limit that we have currently in the United States for drinking water. So we can take some solace that we probably have a safe level there. It turns out the changes that we see below 0.1 micromolar are considered adaptive. Essentially, the cell has to figure out a way to deal with the arsenic and get rid of it. Adaptive changes, there's no toxic response, no adverse response. Between 0.1 and 10 micromolar, you have various toxicities that you're dealing with. If you get above 10 micromolar, you're basically just going to wipe out the cells instantly. It's totally lethal. More importantly is 10 micromolar is almost unachievable in the whole animal, including the whole human, no matter how much inorganic arsenic you take. The reason for that is that once you reach that level, you're dead. So if we look at that, we then say, okay, well, inorganic arsenic leads to the formation of these trivalent arsenicals, the arsenite, MMA3, DMA3. They interact with sulfhydryl groups in the different cells, and depending on what the critical sulfhydryl group is, you have a threshold response, and you generate a non-cancer biological effect. And I want to emphasize that. Arsenic itself does not produce a cancer directly. It produces a precancerous effect. So for in the skin, it produces arseniasis, and you'll notice that that involves inflammation and toxicity, hyperplasia. In the lung, it produces bronchitis, which is also an inflammatory response with regenerative proliferation. And in the bladder, it produces toxicity and regenerative proliferation. The effect of the arsenic is a non-cancer biological effect. It's just that for some tissues, mainly epithelial tissues, these non-cancer effects eventually lead to the generation of cancer because of prolonged regenerative proliferation. Now there's other effects such as the cardiovascular, diabetes, whatever, that are non-cancer toxicities, but essentially it's the same process here that leads to this. So one can then look at the risk assessment for the mode of action for cancer and non-cancer together. So they both involve ingestion of arsenic, conversion to the trivalents, reaction with critical sulfhydryl groups, and then a toxic response. Now if it's epithelial, you get a toxicity with regeneration and ultimately tumors. If it's non-epithelial, you get other types of toxicity, and you get the other non-cancer effects. So we can really approach the overall risk assessment of arsenic for cancer and non-cancer absolutely the same way, which I think would be very useful. Now, the big question then comes down to is how are we going to extrapolate from the high exposure levels that we can easily demonstrate effects, whether it's cancer or non-cancer, to the lower exposure levels that for the most part you and I are exposed to, and estimate what our risk is. And this is where the modelers come in, and I think should be based on basic mode of action considerations rather than just drawing straight lines. I can if you have 2 dots or more, I can draw a straight line and I can tell you what the statistical fit to it is. The problem is it might not have any biological relevance. Now on the drug side of FDA, they are constantly and without exception dealing with the issue of thresholds. I mean, pharmacology and drugs is a threshold phenomenon. Well, on the food side, we have to educate them that I think that's exactly the same process going on. Rather than defaulting to a linear non-threshold risk assessment, my guess is, and all the biology suggests, that the mechanism of the mode of action for arsenic involves a nonlinear threshold phenomenon, and I think we can actually come up with an estimate of what that threshold will be in the human population. So with that, I'd like to conclude that we know what the mode of action is for carcinogenesis. It's nonlinear, it involves a threshold, it involves cytotoxicity and regeneration, and that we can apply the same mode of action to the risk assessment for cancer and non-cancer endpoints. So with that, I'll entertain any questions you have.